I lead the Integrated Circuits and Bioengineering Laboratory in the Electrical and Computer Engineering Department. Our research is in developing integrated circuits for interfacing them with biosystems. For example, we develop chips that we interface with cell cultures, and our goal is to understand how these microbiosystems work and also to understand how they work in the presence of drugs or how they work in disease states. We foresee it having strong impact in the pharmaceutical industry. For example, we develop systems that can monitor cells without labeling them, without staining them, and being able to do that reduces your cost of sample preparation significantly. And we use small analysis volumes, so that means for a given batch, we can get a lot more data at a fraction of the cost. So our systems will revolutionize how some tests are made, some assays are conducted, and they will help speed up the process of drug discovery. Our research is at a pretty high technology readiness level. We have working prototypes. We routinely conduct assays over several days to weeks. And we are hoping that at this point in time, we can partner with a pharmaceutical company to look at how our research can be translated into a product. So one of the biggest challenges that I've experienced myself when I was in industry was the lack of talent. Meaning this is a very highly interdisciplinary field, and it's very hard to find folks that are competent in, for example, biology, and then device engineering, and then technology integration. And it's unusual to have all these skill sets into one person. For example, I think it helps to look at a student who comes in to my group day one, and they're from an electrical engineering background most of the time. And they have some interest in biology and biomedical engineering and so forth. And then I start talking about cell culture and cell adhesion, and they get worried. They're like, that's not my training. I don't understand how to do this. That's day one, but by day three, they'll tell you every single thing there is to know about cell culture. So I think to me, that's the most exciting part is, broadening your horizon by, learning different things and putting it all together to address a very complex set of applications. To me, that's the most exciting part, the highly multidisciplinary nature of the work.